Welcome to Antigen Recognition by Lymphocytes module. In this talk, we're going to learn about the B cell receptor. We will first look at the structure of the B cell receptor and the T cell receptor, and then learn about different types of immunoglobulins and the different parts of the immunoglobulins. Let us begin with a summary of innate versus adaptive immunity. As we learned previously, innate immunity is quick, is fast, and is the first defense that plays a role in controlling pathogens that exhibit certain molecular patterns. However, innate immunity a lot of times is not enough for the efficient removal of a wide range of pathogens, and for that, an adaptive immune response is required. The part of the adaptive immune response are the lymphocytes, which are able to recognize a wide variety of antigens that are present on all kinds of microorganisms. Just to summarize, an antigen is a molecule or part of a molecule that is recognized by special recognition proteins on these lymphocytes. And in the case of a B lymphocyte, this special recognition protein is the B cell receptor, while in the T lymphocyte, this special recognition protein is the T cell receptor. Let us now look at the BCR and the TCR side by side. So the BCR is the B cell receptor, and the TCR, which is the T cell receptor, they both show structural similarities. Now the B cell receptor is the one that is found on B cells and is also called as the immunoglobulin. It is able to recognize and bind to antigens present on pathogens. The T cell receptor, on the other hand, is present on T lymphocytes, and it recognizes and binds to short peptide fragments that come from protein antigens. So it's important to note that the B cell receptor can recognize antigens that don't necessarily have to be proteins. However, the T cell receptor recognizes and binds only to parts of protein antigens. Let us first focus on the B cell receptor or the immunoglobulins. Now do remember that antibodies and immunoglobulins often refer to the same kind of molecule. There are 5 classes of immunoglobulins that our bodies make, and they're all listed in this picture below. So we have the IgG, IgE, IgD, and the Ig in this case stands for immunoglobulin. We also have multimeric immunoglobulins like IgM and IgA. If you focus on the black box, you can see that IgM is made up of 5 immunoglobulins that are linked together, and hence it is a pentameric structure. While in IgA, we have 2 antibodies that are bound together to form a dimeric structure. All of these immunoglobulins have their own specific functions and features that we will learn as we go through the course. Let us look at the basic structure of the antibody. Now, each antibody is made up of 2 different but identical kinds of polypeptides. If you focus on the figure, we can see this Y-shaped structure that is formed by the antibody. Let us look at the different parts of it. So there are 2 heavy chains, and they have been outlined in those black structures, and they are the longer lines that we can see. Every antibody will also have 2 light chains that are denoted in the figure right now. Now, when we focus on those heavy chains, those 2 heavy chains that are part of the antibody are identical to each other, and they are linked together by disulfide bonds that keeps them closer to each other. And the disulfide bond is shown there in that smaller box. Now, each heavy chain is linked to a light chain as shown in the figure, and that also occurs through a disulfide bond. So right now we're looking to the left side of the figure where we have 1 heavy chain bond to 1 light chain, and the same thing is happening on the other side of the antibody too. And now we can see in the smaller boxes where the disulfide bonds are present that are linking each heavy chain to a light chain. These disulfide bonds help the antibody stay intact and not fall apart. So as you can see, every antibody is made up of 4 polypeptides, which are 2 heavy chains and 2 light chains. In the case of humans, there are two types of light chains that are present. So in the figure, we can see where the light chains are, and the two different types of light chains that can be present are the lambda or the kappa light chain. Now, each immunoglobulin will have either two lambda chains or two kappa chains, but never can you have both, which means we will never find an immunoglobulin that will have one lambda and one kappa chain. So either both will be lambda or both will be kappa. However, there is a kappa to lambda ratio, which is that most of the antibodies will have the kappa light chains, whereas some of them will have the lambda. And so about 2/3 of our antibodies have the kappa light chains, while 1/3 have the lambda light chain. The way we have two different types of light chains, we actually have 5 different types of heavy chains, and they are denoted by the Greek alphabets mu, gamma, delta, epsilon, and alpha. Now, based on the type of the heavy chain that is present, we have our 5 different types of antibodies that we saw in a previous figure. Thus, we have our IgM that has the mu heavy chain, IgG has the gamma heavy chain, IgD has the delta heavy chain, IgE has the epsilon heavy chain, whereas IgA has the alpha heavy chain. Thus, the heavy chain determines the kind of immunoglobulin. When we look into the structural aspects of these chains, we can see that both the heavy and the light chain show repeated domains, which are called as the immunoglobulin domains. If we look at the heavy chains on one part of the antibody, here we can see that there are 4 immunoglobulin domains that are being shown in these black circles. So we can see that for one heavy chain, and similarly we will have the 4 immunoglobulin domains for the other heavy chain. In the case of some immunoglobulins, instead of 4, you may have 5 immunoglobulin domains in the heavy chain. The light chains always will have 2 immunoglobulin domains, and the 2 immunoglobulin domains of the light chain are shown for one of the light chains in the black circles, and we can see the similar 2 immunoglobulin domains on the other light chain of the immunoglobulin. Now, the amino acid sequence of the first 110 amino acids, which corresponds to the first immunoglobulin domain, which is shown in the figure in the black circles, shows variation. So different antibodies have a different amino acid sequence in that variable region. Right now, in the black circles, we are seeing the variable region or the variable domain for the light chain that is called as VL. Similarly, the heavy chain also will have that variable domain, which we call as VH. Now, the V regions of the heavy chain and the light chain, as you can see in the figure, are close to each other in both arms of the Y. And hence, they are the ones that actually determine the antigen binding specificity. The remaining immunoglobulin domains that we can see as a whole bunch of black circles in this figure are the constant domains, and they don't show a lot of variability in their amino acid sequence. Hence, they're also called as C domains, and the constant domain for the light chain is called as CL, whereas the constant domain for the heavy chains is called CH. When antibodies were discovered, it wasn't known that which part of the antibody could recognize the antigen and which part of the antibody was able to bind to other cells. And so for that, proteolytic cleavage experiments were performed. And based on those experiments, it was determined that the arms of that Y structure of the antibody that is shown in those black circles are the region that will bind to the antigens, whereas the base of that Y or the stalk of the Y will actually bind to receptors that are present on our innate immune cells, as well as that is the part that can actually bind to the complement, for example, the C1q. Now, these antibodies were subjected to proteolytic cleavage to form distinct fragments that had an associated function. So we're first going to look at what happens if we subject the antibodies to proteolytic cleavage with the protease papain. And as you can see in the figure, when the antibody is subjected to papain, it actually is able to be broken into multiple fragments. There are two fragments that are formed that are showed in the black circles that tend to be identical to each other, and they were called as the Fab fragments because it was the fragment that has the antigen binding site, or FAB, or Fab fragment. So by cleaving the immunoglobulin with papain, we were able to see two Fab fragments, and there was one fragment that was called as the FC fragment. And the reason it's called as FC is because it was the fragment that was easily crystallizable. And so, as you know, proteins can be crystallized to find their three-dimensional structure. And when we looked at the different fragments formed in the case of the immunoglobulin, the FC fragment was easily crystallized, and hence it was given the name FC. Similarly, experiments were performed by cleaving the antibody with other proteases like, in this example, pepsin. Now, in the case of pepsin, its recognition site on the antibody is different, and hence the pattern of cleavage is different. So in the case of pepsin, it cleaves the immunoglobulin at the C terminus of the disulfide bond that holds the heavy chains together, and that is shown in the figure in the right where you can see the red arrows. The result of pepsin cleavage was you would form one fragment which was called as the Fab'2 fragment. It's called as Fab'2 because it's a fragment that has two antigen binding sites that are held together by disulfide bonds. The Fab'2 fragment has the ability to bind to the antigen. However, because it lacks that FC region, it cannot bind to complement or to receptors present on innate immune cells. The remaining fragment was degraded into further smaller fragments, and the largest of these fragments was called as PFC prime. Thus, by subjecting the immunoglobulins to different types of proteases, people were able to elucidate the functions of the different parts of the antibody. With this, we come to the end of our talk, where we learned about the B-cell receptor and different types of immunoglobulins as well as their composition.